Students, today we will discuss another very important chapter in your biochemistry syllabus, that is nucleic acids. So under the section we will discuss regarding the definition of different type of nucleotides and what are the different type of purine and pyrimidine bases. We will discuss about nucleoside and nucleotide with proper examples. We have to study the structure of DNA, and that was proposed by the two famous scientists named Watson and Crick. And at the same time we also discuss regarding the RNA and different type of RNA and their functions. So this is the content we are going to cover according to the syllabus present in the diploma course. So first of all, we discuss regarding nucleic acids. So whenever we are talking about nucleic acid, that signifies that it must be related to nucleus. So you know that this is our cell. The cell may be plant cell or animal cell. All the eukaryotic cell contain a nucleus, and inside the nucleus some acidic material are present, which is popularly known as nucleic acids. So under the nucleic acid, the examples are your DNA and RNA. You have already known about DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for your ribonucleic acid. Both DNA and RNA are example of nucleic acids. Okay, and the DNA and RNA are the building block of genetic material. That means the genes are present in DNA and RNA. In most of the cases, DNA is the main building block material, a building block of genetic material. But in certain viruses, RNA also act as genetic materials. A genetic block act as building block of genetic materials. Okay, so now the question that what type of organism that contain the RNA, and that RNA is coming under the that RNA contains genetic material, that is the virus. So you know that virus are different type, DNA virus, RNA virus, because virus is such a organism which do not contain both DNA RNA, either DNA present or RNA present. So on that basis, viruses are divided into different categories, like your RNA virus and DNA virus. Clear? RNA virus means this contain only the RNA. DNA virus, it contain only the DNA. In the your virus, there is no both DNA and RNA. No, there is not presence of both DNA and RNA. Okay, so basically, in case of eukaryotic cell, but in case of your other organism, RNA actually help in your act as a messenger, and they help in protein synthesis. So generally we can say that DNA is contain the gene and RNA helpful in protein synthesis. Clear? So we have to think of the people. Look at the picture. In the picture of Johannes Friedrich, who actually the first Swiss physician and biologist who discovered about nucleic acid, who was the first person who isolated the nucleic acid from the cell or from the nucleus in 1869. Okay, so this picture, look at the picture here. This is the typical diagram of a cell, and the cell contain your nucleus, the nucleus, nucleoplasm, endoplasmic reticulums, ribosomes, nuclear pores. The gap between the membrane is nuclear pore, and the cover is called nuclear membranes, like that ones. So the genetic material or the nucleic acid present inside the nucleus. Okay, so as again I repeat here, as they found in the nucleus, so they are called your nucleic acids. Okay, now basically nucleic acid two type, DNA and RNA. So first we discuss about DNA, next we go to the RNA. So DNA is such a genetic material which actually contain or carry the genes. Okay, and DNA is made of two polynucleotide chain coil around each other. That we will discuss in the coming slides. Okay, so DNA is a polynucleotide and they are polymer of nucleotide. That means DNA are made of many nucleotides and that is called polynucleotides. Okay, like nucleotide 1, nucleotide 2, nucleotide 3, nucleotide 4, like that ones. They combine form a thing. It's like a garland. Okay, so each nucleotide are joined to each other and make a chain, and that chain is called polynucleotides. Okay, interestingly, DNA consists of two polynucleotide chains which move around each others. Okay, so remember that DNA is having two polynucleotide chains, where RNA having a single polynucleotide chains. Clear? All of you clear my point? Again I repeat here. I want to repeat again. The DNA is the double polynucleotide chains which coil around each other. Okay, but RNA is single polynucleotide chains. Okay, now we discuss about the structure of your polynucleotide chains. Okay, so polynucleotides are the polymer of nucleotide. That means many nucleotides combined to form polynucleotides. Okay, both DNA and RNA are polynucleotides. Okay, and each nucleotide is formed by three things. One is nitrogen base, then pentose sugar, then phosphate group. These three combinedly called as nucleotides is known as nucleotides. Okay, but whenever the nucleotide do not contain the phosphorus, then it is called nucleoside. Okay, so do not confuse about this one. Nucleotide consists of three things: nitrogen base, pentose sugar, phosphate group. But nucleoside contain nitrogen base and pentose sugar. There is no phosphate group. Okay. Now, as I told earlier, DNA is having the three things. Okay, it's having the nitrogen bases. So now the nitrogen bases again two type. One is called purine bases, second pyrimidine bases. Okay, purine having again two type of purines: adenine bases, guanine bases. Pyrimidine having cytosine, thiamine, and uracil. So lower one are the pyrimidine bases and upper one is the purine basis. तो purine के अंदर कौन आया? adenine आया, guanine आया, pyrimidine में cytosine, thiamine and uracils. Okay, but however, in each your nucleic acid, only out of 5, only 4 nitrogen base may present. Like in case of DNA, it contains adenine, guanine, cytosine and thiamine. In case of RNA, it contains adenine, guanine, cytosine and uracil. Okay, so the difference between DNA and RNA on the basis of nitrogen base, that DNA contain thiamine, RNA contained your uracil. That means there is no uracil in DNA and there is no thiamine in the RNA. Okay, so now look at the picture. This is the two rings. Look at the picture. The two polynucleotide chain are coiled around each other. Okay, and this coiling actually occur due to the linkage or bond. What bond? The type of bond which cause two polynuclear chain close to each other, that is your hydrogen bond. Remember that, hydrogen bond. Okay, initially they present two nucleotides, but due to the hydrogen bond, what happens that now it coil around each other like this ones. Okay, apart from the hydrogen bond, other bonds are present. Okay, that need to discuss. Now, what is the first one? That your N glycoside linkage. What is this thing? Now, this is the type of bond which occur between the fast carbon of the pentose sugar and the nitrogen base. So, this bond is called N glycoside bond. Because if you do the numbering 1, 2, 3, 4, 5, तो 1 कार्बन के पास एक बॉन्ड फॉर्मेशन होगा. ओके, किसके साथ होगा? नाइट्रोजन बेस में होगा. नाइट्रोजन मतलब एडेनिन हो सकते हैं, ग्वानिन हो सकता है, साइटोसिन हो सकता है, थायमिन हो सकता है. ओके, so this bond is called N glycoside bonds. Okay, but the bending occur, these all are your hydrogen bond. These all are hydrogen bond, and hydrogen bond occur between two nitrogen bases, just like A double bond T and G triple bond C. That means when the adenine bond with the thymine, adenine of one chain, okay, make bond with the thymine with the another chain. Similarly, guanine of one chain make bond with the cytosine of another chains. Okay, so this bond, either double bond or triple bond, are the hydrogen bond. So due to these hydrogen formations, now the two chains are coil around each others like this ones. Okay, then this is the nucleoside. As I told, nucleoside do not contain the phosphate group. So nucleosides of DNA are like deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymine. Okay, similar nucleotide in RNA, adenosine, guanosine, cytidine and uridine. Same thing is that. In this both nucleoside DNA and RNA, there is no phosphorus. There is no phosphorus attached. No phosphorus group attached. Okay. Now look this next slide. इस slide में लिखा है कि देखो, now one position of pentose sugar, because pentose middle one, bind with your nitrogen base, either adenine, guanine, cytosine or thymine. Okay, and fifth position also bond to a phosphate group. Okay, similarly the 3 carbon also combine with the phosphate group. So generally in the pentose sugar, 3 carbon and 5 carbon, they bind to phosphate group. Okay, but now in the DNA, what is happening? That look at the position in the RNA and the DNA. The difference in the DNA RNA on the basis of pentose sugar, that DNA contain your deoxyribosugar, RNA contain ribosugar. Both are the pentose sugar, but different that in case of RNA, in case of DNA, there is no OH group oxygen present in the second carbon. No oxygen presents. Okay, are you getting point? But in RNA, each nucleotide has additional OH group at 2 positions. But in DNA, there is no OH group. But RNA, there is OH group. Okay. Now, we go to the structure of DNA. The structure of DNA was proposed by many scientists, but finally, one of the famous achievement by the world scientists, that is proposed by 2 famous scientists. One is called Watson and Crick. Watson Crick, that means James Watson and Francis Crick from America. They proposed the structure of DNA. Okay, that is double helical structure model of DNA. because according to James and Watson and Crick, DNA are double helical structure, okay, and in which two polynucleotide chain move opposite to each other and coil around each other. This was the proposed model, first proposed model that was proposed by Watson and Crick. Later on, it was accepted by many people, and for this, in 1953, these two combo got the Nobel Prize. And they, another important, we do not forget that contribution of another two scientists, that is your Wilkins and Rosalind Franklin, the two scientists who actually gave the structure of DNA by XRD, that is on the XRD X-ray diffraction data given by these two scientists named Wilkins and Franklin. Okay, on the basis of the structure, XRD structure, Watson and Crick proposed the models. So here 3 or 4 pictures are there that need to remember. One is the Frederick mixture, who was the first person who separated the nucleic acid. Watson Crick, the first person who proposed the DNA double helical structure. Then Franklin and your Wilkins, the two scientists who actually gave the structure of DNA by XRD. Okay, that means they gave first step the data by using the XRD. So now if you discuss about the structure of DNA having two polynucleotide chain, as I told, this is one polynucleotide chain, this is two polynucleotide chains, okay, and they coil around each others. Now second important point, that the pitch of the DNA helix equal to 3.4 angstrom, 3.4 nanometer or 34 angstrom. That means from here to here, the total distance equal to 3.4 nanometer. If you convert into another unit, that is 34 angstrom. Next important thing he described that in one pitch, okay, the total number of base pair equal to 10. Base pair means here A double bond T, G triple bond C. So this is one base pair and this is another base pair. Clear? So two base pair are there. Okay, so this says that in one pitch, okay, but in one pitch the total number of base pair equal to 10. And if we measure the distance between the two successive nucleotides, suppose two successive base pair, this is one base pair, this is another base pair, so the distance between the equal to 3.4 divided by 10 equal to 0.34 angstrom, 0.34 nanometer or 3.4 angstroms. Okay, then what they say, the backbone of the DNA is formed due to presence of sugar and phosphate. Okay, backbones, because due to presence of sugar, which help to hold the nitrogen bases. Without the help of the sugar, there is also no existence of your DNA. Okay, and another important thing, that in the DNA, two polynucleotide chain are moving opposite to each other. That is called anti-parallel polarity. That means one is going this direction, another going this direction. That is called anti-parallel polarity. So that is called as your 3' to 5', and this is your 5' to 3'. Okay, look at the picture. As I earlier told, A always make bond with T, 2 hydrogen bond, C or G triple bond with the G, and hitting 3 hydrogen bonds. So remember that purine is make opposite bond to with pyrimidines. Okay, that's why adenine always make with bond of thymine, and guanine always makes triple bond with the cytosines. Okay, adenine the purine base, and thymine the pyrimidine bases, guanine the purine base, cytosine the pyrimidine bases. Okay, this thing. Next one another rules that was given by the Erwin Chargaffs. According to him, in DNA, the proportion of adenine is equal to thymine and the proportion of guanine is equal to cytosines. So therefore, we can write that adenine plus guanine equal to cytosine plus thymine. That means total purine equal to total pyrimidine. एक डीएन में जितना प्यूरिन बेसिस है उतना ही पिरामिड बेसिस रहेगा और जितना ही पिरामिड बेसिस है उतना प्यूरिन बेस रहेगा. इफ आई you can change, then a plus g plus t plus c is equal to 1. The ratio, that means the ratio between the combination of the adenine and guanine to the combination of cytosine thymine equal to 1. Okay, next we will discuss very briefly, that is your RNA. Okay, the RNA is called double helical, single helical structure, single standard structure, and they the main role of RNA in most of cases help in protein synthesis or directly indirectly involved in the synthesis of protein by making of proteins. Okay, there are three types of RNA. One is called mRNA or messenger RNA. Second, the tRNA, transport RNA. Third one, the ribosomal RNA, rRNA. Okay, three types of RNA are found. tRNA, which is also called as soluble RNA, and transport RNA actually carry the information from DNA to the cytoplasms. Okay, look at the picture. This is the structure of the tRNA, transport RNA, and the transport RNA is responsible for to transporting the amino acid to the site of protein synthesis, that means to the site of ribosomes. Okay, so this DNA actually Francis Crick, same scientist who got the Nobel Prize for the DNA structure, he was the first person who postulated the presence of the adapter molecule that can read the code and link with the amino acids. So tRNA is called adapter molecule because it has anticodon. This is the anticodons. Okay, tRNA have an anticodon, and your mRNA, which coming from the your nucleus, having the codons. Okay, so codon and anticodon, whenever the match, then only it will release the amino acids. Okay, and this tRNA having different structure, different site. This is called your amino acid site, where amino acids are attached here. This is called ribosome loop, binding loop. That means here the tRNA bind to ribosome for protein synthesis, and this is called enzyme binding loop. This is actually the main site of the rtRNA which bind to the enzyme. Okay, and this is called your anticodon which bind to the codon. So whenever the codon and anticodon match with each other, at that time there is a release of the DNA, the release of amino acid from the tRNA. Okay, then I told earlier that, and the same thing, the tRNA particularly present in the cytoplasm, helping transporting the amino acid from cytoplasm to the ribosome. And mRNA, which carry the message from DNA from nucleus to the cytoplasm, and rRNA, ribosomal RNA, which indirectly help in protein synthesis. Okay, and they are found sequestered more abundantly in nucleus that present inside the nucleus. Okay, look the structure here. This is the binding site of the amino acid, serine, tyrosine, etc. This is your anticodon, like UCA anticodon. So codon होगा AGU opposite, because I told that A double bond T, G triple bond C. So when it is A, that means it is T. यहाँ पे T रहेगा, वहाँ पे G, यहाँ पे C रहेगा. Okay, this thing. यहाँ पे U, okay, यहाँ पे भी वहाँ क्या रहेगा, A रहेगा. Okay, because U present in the thing RNA. Okay, so this is the codon, anticodon. When codon and anticodon match, then only the tRNA release the amino acids. So like amino acids join to each other to form the polypeptide chain. Polypeptide chain join to form from the proteins. Okay. Okay. So this is a short video, quick video for the nucleotides And the question, the probable question you can get that the write down the structure of DNA, most important. Maybe you get the question, long question. Okay, write down the structure of DNA. And sometime also you can question about your Chargaff's rules. Okay, this Chargaff's rule may also be important. This one, Chargaff's rule, that also important. And very little question will come like what is who is the first person who identified the who isolated the nucleic acid from the cell, like that ones. Okay, and what is the how many what type of bonds are formed in the DNA, like that one you can get the questions. Okay, thank you very much, have a nice day.